So if we're talking about innate versus adaptive immunity, the two main themes of our innate immune systems is that it's non-specific, and then it's going to be the same magnitude of response each time. So this is things like your complement system and your phagocytes that are going to kick in immediately when you encounter a pathogen, and every time you encounter that same pathogen, it's going to be the same magnitude each time. Same tricks that those cells have. And we say it's non-specific, so our innate immune system can recognize pathogens, right? Because they have pattern recognition receptors. So they have TLRs and RLRs and NLRs and CLRs, so they can recognize broad motifs that pathogens have. But they can't tell an influenza virus maybe from a coronavirus, right? Or they maybe can't tell two different kinds of gram-negative bacteria apart, because they're just going to be recognizing really broad molecular patterns that pathogens have, and they won't know this is the strain of flu that you had 7 months ago, because they can't recognize very specific pathogens. Whereas the adaptive immune system is highly specific to each unique pattern, each unique pathogen. So it recognizes the exact pathogen that has infected you in the past, regardless of its broad molecular signatures that it has. The innate immune system will still recognize those. But to mount these highly specific responses that give you your immunological memory, you need your adaptive immune system. And also in parallel, the adaptive immune system takes time to develop after the initial pathogen encounter, so it's slower, because it takes time to form that very specific response against that pathogen. But then on a second encounter of that pathogen, it acts immediately and most of the time before you even have symptoms. So we just say you're immune to that because you've seen it before. So adaptive immune system is very specific and responsible for your memory. Okay, so the first thing, the defense systems of prokaryotes work similarly. So they kind of have innate immune mechanisms that are non-specific and have the same magnitude of response each time. And they also have ways that they can mount responses to very, very specific pathogens that they've seen in the past that take longer to develop the first time, but then upon a second encounter are strong. So first we're going to talk about innate immunity in prokaryotes. And so this is from that SEED review that I posted by Kimberly Seed, which kind of goes through step by step all these different stages in phage infection of bacteria, and then outlines how bacteria try to block it at each phase. So that's a good one to review. So this is just a review of what we talked about in the first 10 or so minutes of class. And so as we said, the first step to phage infection is adsorption, or the phage attachment to a receptor on the surface of the bacterium. So how do bacteria block this from happening? And so one of the ways, so in this cartoon here, if this is the phage and this kind of green banana on the surface of the bacteria is the phage receptor. So the phage needs to bind to this specific receptor to get into this bacterium. One of the things that bacteria do is shed membrane vesicles. And so if this is a membrane protein that's acting as a receptor, this will be kind of producing decoys of the receptor out in the environment, so that these decoys can then kind of soak up all the phage in the environment so that fewer of them are likely to get to the surface of the bacterium. So that's one way that bacteria evade absorption. Another thing they can do, kind of similar to what our cells do, is they can conceal their receptor. So they can actually form a capsule on the surface of the bacteria, which is composed of polysaccharides, so that this kind of obscures availability of that protein receptor for the phage to be able to bind to. And interestingly, we've met bacterial capsules before. These are actually a way that bacteria can evade our immune system. So polysaccharide capsules that bacteria will form are ways to both avoid host immunity, but also ways to avoid phages accessing phage receptors on the surface of the bacterium. Bacteria can also mutate these receptors. So let's say the green banana was the receptor that this phage attaches to, so maybe there's going to be a point mutation that's going to change the morphology of this protein, which will no longer be able to bind to the phage, so the phage won't have access. And bacteria also do something called phase variation. which is kind of similar to antigenic variation that we've talked about. But basically the bacteria may have a whole family of surface proteins that have a very similar function. And so they kind of, but maybe phages use one of those proteins to infect the cells. So the bacteria can kind of switch up the expression of those proteins at different times throughout their life cycles, so that at certain points throughout their life cycles they're resistant to the phage. So phase variation again is something that may be useful to the bacteria to evade mammalian immune systems, because let's say that you're in a host that's produced antibodies against you. So then it may be beneficial to be switching up your surface proteins so that you can evade recognition of antibodies. But another reason that this is likely beneficial in bacteria is because this can downregulate certain phage receptors. So phase variation is another strategy that bacteria use that has two advantages in surviving. Okay, so next we're going to talk about restriction modification systems. So these act after the phage has absorbed, if it's bypassed all of the bacterial defense mechanisms that prevent that, and does actually attach and inject its DNA. There are mechanisms that the bacteria can use to actually chop up that DNA so that it doesn't actually go into the cells and form a productive infection. And these are called restriction modification systems. And so you may have, if you've ever taken a molecular biology class or worked in a molecular biology lab, you've probably heard of restriction enzymes, right? So restriction enzymes actually come from, they're naturally made by bacteria as part of the bacteria's innate immune system. And we as researchers have figured out how to exploit that to use them in the lab to cut up DNA at certain sequences. So restriction enzymes are enzymes that recognize palindromic sequences. And there are many, many, many restriction enzymes, hundreds I think, that have been identified. And each of them recognizes a different specific sequence. And all of these sequences are palindromes, the top strand versus the bottom strand. And so the restriction enzymes can recognize these sequences and then cleave at those sequences. So this is a good way of chopping up incoming phage DNA. And so the reason that these restriction enzymes don't just chop up the bacterial DNA that may happen to have these sequences throughout it is because these are because this is a restriction modification system. And so the restriction part, the restriction enzyme, is only the first half of it. So the second half is the modification part. And the modification part of the system is these methyltransferases that bacteria have. So bacteria have methyltransferases that methylate self-DNA at these regions that the enzymes are going to cut at. And when the DNA is methylated, the restriction enzymes will have less access to be able to cut. So in this way, the bacteria can protect their own DNA at those sequences that the restriction enzyme is going to cut at, and cleave incoming phage DNA where these sequences are. And so also restriction enzyme, we can also use the word restriction endonuclease. So these are nucleases inside the bacterial cell. So enzymes that cleave DNA inside the bacterial cell. So you can imagine that one thing that phages might then evolve to do in this arms race is start to methylate some of these sequences, right? So in studying the evolutionary record, biologists have seen this, and bacteria have then adapted additional restriction enzymes that can recognize methylated sequences. So there's a little bit of back and forth with the evolution of these as well.